We previously discussed differential expression methods for the microarray setting. In this module, we will focus on differential expression methods for RNA sequencing data. To review differential expression analysis, we are interested in identifying genes that are over or underexpressed in one condition compared to another, or perhaps we are interested in identifying genes with expression levels significantly associated with a continuous phenotype, such as blood pressure. For each gene, we apply some inferential procedure, such as a two-sample t-test or its nonparametric counterpart, the Wilcoxon rank sum test, to obtain p-values. Or if you have multiple groups, for each gene, we fit a linear regression model, which is equivalent to analysis of variance, to obtain p-values. Then we adjust the resulting p-values by either a family-wise error rate method, or we use them to obtain the false discovery rate. Likewise, if you have a continuous predictor for each gene, we fit a linear regression model, extract the p-values, then adjust them using these methods. Alternatively, the limma bioconductor package can be used, which fits a linear model and then uses Bayesian methods to get maximum a posterior estimates of standard error, resulting in moderated test statistics. The TCGA BioLinks bioconductor package facilitates the Cancer Genome Atlas at open access data retrieval. The summarized experiment class in the bioconductor package of the same name is used to store rectangular matrices of experimental results, which are commonly produced by sequencing and microarray experiments. Here we download gene expression count data from RNA-Sequencing assays for the glioblastoma samples from the Cancer Genome Atlas project. Here is a snapshot of the number of reads for 5 genes for 2 samples. The phenotypic data in the summarized experiment object can be extracted using the call data function. One phenotypic variable that may be of interest is sample type, which is either primary tumor, recurrent tumor, or solid normal tissue. As we saw in the last module, the limma bioconductor package is used in the analysis of microarray data, but it can also be used to analyze RNA sequencing data. This is convenient because applying linear models allows us to flexibly perform our analysis given the study design. When using limma to analyze RNA sequencing data, read counts are converted to log base 2 counts per million, and the mean variance relationship is modeled either with precision weights, called Voom normalization, or with an empirical Bayes prior trend, called limma trend. Voom normalization is recommended when the library sizes are variable among the samples. The limma trend approach is a simpler method that can be used when the library size is consistent among samples. Then the normalized gene expression data are analyzed as if they were microarray data. That is, a linear model is fit, contrasts are constructed if they are of interest, and empirical Bayes is used to perform moderated gene-specific tests. The limma package includes many useful functions such as MAplot, Venn diagram, and volcano plotting functions. Prior to the analysis, often the gene count matrix is filtered to remove genes with 0 counts across samples or genes with very low counts across samples. Then read counts are converted to log base 2 counts per million, and the mean variance relationship is modeled using Voom normalization. Then we proceed as if the data were from a microarray. Here's a quick review of our linear regression model, where for gene J in samples 1 to n, we have log base 2 transform Voom normalized gene expression data represented by Y. Note that the boldface font indicates we have a vector or matrix. The linear model for Y is that its expected value is x times beta. Here, x is a full rank design matrix representing the conditions or groups of interest. Beta is the vector, which is the same length as the number of columns in our design matrix X. The coefficients beta sub j reflect the association between our covariate and gene expression. When fitting a linear regression model using ordinary least squares, the variance for a specific gene will be constant. To formalize the concept of the X design matrix, suppose we have 6 samples, subjects 1 and 2 belong to group 1, subjects 3 and 4 belong to group 2, and subjects 5 and 6 belong to group 3. The columns are indicators of biological conditions, sometimes called dummy variables. Notice that beta is the same length as the number of columns in our design matrix X, where beta sub jk is the expected expression level of gene J in group K. Returning to the TCGA glioblastoma data, recall that there are 3 groups, so we will define the design matrix using the model.matrix function, where -1 indicates we do not want to include an intercept. We can see the format of our design matrix and verify the number of subjects in each group matches our result by applying the column sums function to our design matrix. In this example, we have 3 groups, so we are likely interested in forming pairwise contrasts, which are linear combinations of our coefficients betas. The linear model provides coefficient estimates and an estimate of our residual variance, sigma squared, which is given by S squared. Given a contrast matrix C and our coefficient estimates stored in our vector beta, we can form the comparisons of interest. Given the estimated variance-covariance matrix of our model parameter estimates, we have the variance-covariance matrix of the contrast matrix. The square roots of the diagonal elements of this matrix are then the unscaled standard deviations. If we are interested in all pairwise comparisons for the 3 groups, our contrast matrix would be structured like this. That is, we want to compare each of the 2 groups. So column 1 is 1 minus 1 and 0, and this is group 1 minus group 2. In column 2, we have 0, 1, and minus 1, and this is group 2 minus group 3. In the third column, we have 1, 0, and minus 1. This is group 1 minus group 3. Therefore, this matrix multiplication involving the contrast matrix yields the expected differences in expression level of gene J comparing the groups, which is the log fold change if the data are log transformed. We have already set up our design matrix X to include indicator variables for each of the 3 sample types, primary, recurrent, and normal. We can now use limma's makeContrast function and specify the paired differences by name and include that those levels are defined in our design matrix. In an ordinary linear regression model, we test contrasts using a t-test. In fact, if there are only 2 groups, then the contrast is 1 minus 1, which is equivalent to the 2-sample t-test assuming equal variance. However, if counts are low or sample size is small, you may underestimate the variance, which could lead to false positives. So the limma package moderates the variance. The empirical Bayes method assumes a prior distribution for sigma squared, the variance, which is a scale chi-squared distribution as represented here. The posterior values of the residual variances are given by S tilde sub j squared, which is a weighted average of S sub j and S sub naught, where D sub j is the residual degrees of freedom for the jth gene. We plug in this updated variance to get our moderated test statistic, which under the null hypothesis follows a t distribution with d naught plus d sub j degrees of freedom. The prior degrees of freedom represent the extra information borrowed from all genes. Besides using moderated t-tests, limma uses empirical Bayes, the probability of gene J being differentially expressed for the mth contrast is given by P sub m. The beta coefficient follows a normal distribution, and these parameters are estimated from the data, and hence we have empirical Bayes in use. We calculate the posterior odds to get the B statistic, which is the log of the posterior odds. If B is greater than or equal to 0, that indicates the gene is more likely to be differentially expressed than non-differentially expressed. Note that if you order the genes by these B statistics, you will not necessarily get the same resulting order as if you order the genes by their p-values. Therefore, it is recommended to use the p-values from the moderated t-tests and estimate the false discovery rate instead of relying on these B statistics. We have already specified our design matrix and set up our contrast matrix. Before we fit our limma model, we transform the count data using Voom. By setting the parameter plot equals true, we get a graph that displays the estimated mean variance trend. Then to perform the differential gene expression analysis, we first fit the linear model, then estimate the contrasts, then get empirical Bayes estimates. Perhaps we are interested in the primary versus recurrent paired comparison. In that case, we can view the top differentially expressed genes using the top table function by specifying we are interested in that specific comparison. Note here that we have sorted by the p-values and that the corresponding B statistics are not always decreasing. We probably want results for all of the genes instead of only the top 10, so we can set the number parameter in top table to infinity. Now we see that there are 17,838 genes identified among the 60,000. When comparing two groups or examining a pairwise contrast, results from the differential gene expression analysis are often graphically displayed in a volcano plot. In a volcano plot, the minus log base 10 of the p-value, alternatively, you could use the FDR, is plotted against the log base 2 fold change. Therefore, points in the top left and top right corners of the plot are often biologically interesting, as they represent genes that have both small p-values and large fold changes. The limma package includes a function for producing a volcano plot, which allows the contrast of interest to be specified. The highlight parameter allows you to identify a selected number of genes that have the smallest p-values. Hence, they also have the smallest FDRs, and their labels are given by the names parameter.